Thanks. Um, can everyone hear me? Is this thing on? Oh, okay. Cool. Hi. So thanks for the introduction. My name is Uthsav. I'm currently a postdoc at the Broad and recently graduated from Ben Raphael's lab. And today I'm really excited to talk to you guys about our new work on modeling spatial gradients and spatial domains in spatial transcriptomics data. So I'm sure some of us know what spatial transcriptomics is, but for those who don't, it's a really exciting new class of technologies where you can at a high level, you can measure gene expression at a spatial resolution. So the way that you can imagine this works is imagine you have a tissue slice, a very thin 2D slice of a tissue. At different spots in this tissue, shown as these kind of like green or maybe it just looks white on the screen, at these circles, you can measure both their spatial coordinates, like the XY location, as well as their gene expression, giving you this gene by spot matrix, which you might see in like single cell analysis, but also the XY spatial coordinates. And so there's been a lot of really exciting new technologies for measuring spatial transcriptomics that differ in sorts of resolution of the data, the type of sequencing or imaging that they use, and so on. So for example, 10x Visium is one technology where these spots are 55 microns, so they contain multiple cells, but you can measure all genes. While MERFISH is an imaging-based technology where each spot is a single cell, but you can measure only a handful of genes. What's cool about spatial transcriptomics is that you can now study how tissues are organized at both the kind of molecular and the spatial level. So one problem that is often solved when looking at spatial transcriptomics data is the analysis of spatial domains. These are kind of regions of a tissue that contain cells with similar cell type or gene expression profiles. And so, for example, here's a tissue from 10x Visium. This is a breast cancer tumor. And if you look at the imaging, you can see that there's sort of like some of these different regions that are different cancer regions. The gene expression data, it can help you in finding these more fine-grained domains. And oftentimes these domains can be distinguished by gene expression. So you often have genes that are kind of high in one domain and low in other domains. And these are sometimes called marker genes. In addition to these kind of marker gene patterns where you have genes that are kind of high or low in different areas, you can also have more subtle patterns of gene expression. And so oftentimes you'll also have genes that exhibit what are called gradients of expression. They kind of continually vary across space. And so, for example, you can have genes with radial gradients that are low in the center and high in the outside of a domain, or you can have genes with other patterns. For example, this gene kind of varies from left to right in this domain. And understanding the spatial gradients of gene expression is really important for understanding many biological processes. So for example, in the tumor microenvironment, this microenvironment can be regulated by certain gradients. And in the brain, cell type heterogeneity is sort of determined by these spatial gradients. However, most spatial transcriptomics algorithms cannot identify both spatial domains and spatial gradients. So on the one hand, you have many algorithms that have been developed over the past several years for finding spatial domains from spatial transcriptomics data. And many of these methods kind of implicitly assume that your gene expression is constant, or at least the average expression is constant inside a domain. And they don't explicitly model spatial variation like gradients within a domain. And this is because at a high level, a lot of methods often will train kind of fancy neural networks like GNNs or VAEs that learn these like low dimensional embeddings, and they'll find domains by clustering these embeddings. But these clustering algorithms don't account for kind of spatial variation inside a domain necessarily. On the other hand, there's actually only a few methods that can even identify spatial gradients of gene expression within domains. And many of these existing methods will require substantial prior knowledge. For example, requiring a manually defined coordinate system or boundary curves of domains. Over the past few years, we recently developed an algorithm that we called GASTON that learns both spatial domains and spatial gradients without this prior knowledge. And at a high level, our algorithm sort of works as follows. If you have spatial transcriptomics data, here I'm showing this fictional kind of tissue that looks like a circle, we train this neural network. And this neural network has this one hidden layer that we call the isodepth. And the analogy that you should think of is this hidden layer is a single node. So at every point in your tissue, it assigns a single number. And this number is analogous to the sort of height or like a potential function on your tissue. And what's interesting is that you can, the gradient of this isodepth coordinate, this sort of height coordinate, is a vector field shown as these arrows, which indicates directions of maximum variation in your gene expression. And we had some theory that we proved that you can actually show that this isodepth coordinate, this like potential function exists under certain mathematical assumptions. And so the way that you go from this topographic map defined by this isodepth coordinate to domains and gradients is that spatial domains we showed are bounded by contours of equal isodepth. So for example, in this kind of circular tissue, you might have these there are 3 domains, and this like blue domain is bounded by one curve that's all points with isodepth equals C2, and this other curve, this green curve, that's all points with isodepth equal to C1, like points of the same height. Similar to how if you're like hiking, mountains might be bounded by like curves of the same height on like a topographic map. And then if you look at gene expression, you can model these spatial gradients by looking at how expression varies along this 1D isodepth curve. And so for example, you might have genes that sort of have a gradient here. For this gene has a kind of radial gradient in this like middle layer. And if you look at how gene expression varies along this 1D coordinate, you would see that it's sort of like flat in the regions where there's no gradient, and it has a large slope where there is a gradient. So these 1D plots also tell you about spatial gradients. And so this is work that came out like last year or 2 years ago. And we were quite excited about this work, but as happens when you like publish a paper, people find ways to break your method. And so actually there's this paper from Jackson et al. From Leo Pachter's group that found that our method Gaston actually performs quite poorly in identifying when you have tissues with very complex spatial domains. So here they had this example of a tissue which has this like checkerboard pattern of these different domains. You can kind of see it here. And then if they compare a bunch of like different methods for finding these domains, and Gaston doesn't do very well. And I don't know, normally people might be a bit disappointed when your method doesn't do well, but I was quite excited when I saw this because I mean, first it means that someone actually like read our paper or at least read the GitHub and tried to run it, which is like more than, I don't know, most people will do. But they also like suggested ways and you can improve this method. And so when we saw this, we started thinking about like, what are the other failure cases of our model? When does it not work? And so in fact, kind of motivated by this checkerboard example, we found an even simpler counterexample to our Gaston model. And so here, consider like a really simple tissue. This tissue just has 2 domains. They're both rectangles. And let's just say there's even one gene. And this one gene has like these 2 gradients. It kind of has a gradient from left to right in this first domain, like along the x-axis. And then it's increasing, and it has a gradient from bottom to top in the y-direction in the other domain. And this is kind of informal, so I won't like kind of present the proof. So we actually were able to prove that for this specific tissue, you can't find a single 1D, like a continuous 1D coordinate that can describe this expression value. And the reason why is that kind of fundamentally for this tissue, what you actually want is you want a different coordinate inside each domain. Like inside this domain, you want a coordinate that kind of goes left to right, which shows you that your gradient direction is like in the x-axis. And then for this like other domain, this blue one, you want a coordinate that sort of goes up to down, up, down. And so this motivated us to kind of relax this assumption that you need a continuous 1D coordinate across your tissue. And so we started thinking about maybe you can learn a piecewise continuous isodepth coordinate, where the pieces correspond to these different domains. And so that, at a very broad level, is our new model. So we developed this model of spatial domains and gradients with a piecewise continuous model of this isodepth coordinate. And we derived an algorithm that we called GASTON-Mix that uses a mixture of experts' deep learning model to find these gradients and domains in terms of this piecewise continuous function. And we show that with this kind of more general model of spatial gradients and spatial domains, we get improved accuracy in various tasks in spatial transcriptomics data. Let's now kind of briefly describe what this model looks like. And so the way that we like to think about gene expression is in terms of functions. This is somewhat different than how it might be presented in other spatial transcriptomics literature. But so the way you should imagine this is that if you have a single gene, I've kind of called it G here, you should imagine that you have a function in space. Fg of xy is a function where at every point xy, it gives you the expression of that gene. And we model this as being piecewise continuous. And so the pieces correspond to these different domains. So for example, if you have this kind of circular tissue here, there's like 3 domains. There's this little circle, there's a triangle, and then there's everything else. And we assume expressions kind of continuous inside each of these domains. And so for example, this gene is only expressed in the circular domain, while this gene is expressed in all 3 domains. This third gene is only expressed in, you can kind of see it, but it's actually like light. It's like white 0 here, kind of non-zero, and then highly expressed in this triangle domain. And so if you have these gene expression values, how can you think about spatial gradients? So there's actually a natural tool from calculus for modeling gradients, which is just kind of the gradient of a function. And so if you recall maybe from like multivariable calculus classes, if you have a function in space, the gradient is a vector field where at every point it tells you the direction that your measurements are like maximally increasing. And so for example, for this fictional gene here, this vector field, these spatial gradients points kind of outward because it's small in the middle and it gets larger as you go outside. Whereas for this gene, and you kind of really have to squint to see it, this gene has a gradient in this triangle domain from left to right. So this vector field points like kind of from left to right, and it has kind of a top to bottom gradient in the other domain. So this vector field, this function, or this vector field points from top to bottom. And then this third gene, it has no gradient. It's constant in every domain. So all of these spatial gradients are the zero vector. Of course, this sounds very nice and simple, but one of the challenges in spatial transcriptomics data is that your data is really sparse. And so in practice, you have 70% or more zeros. And so your data doesn't look very neat, and you instead get pictures like this, where, I don't know, I can't really tell if there's a gradient or not. It's sometimes even hard to tell where the domains even are, like this triangle versus the circle. And so you need, because this data can be quite sparse and have low dynamic range, you need some assumptions. You have to place some assumptions on these functions, these domains, and these gradients in order to learn them from the data. And so at a high level, we show that there's actually two mathematical assumptions you can make that make learning these gradients and domains tractable. And so the first assumption is that all genes share the same direction of these spatial gradients. They all point in the same kind of direction inside each domain. And then there's this kind of vector field, V sub p, that's shared by all gene gradients. And notice that each gene can still have different magnitudes of their gradient, and they could also point in a different sign, but the direction is kind of the same. And then the second assumption that we make is that this vector field is what's called a conservative vector field, meaning that it's the gradient of a scalar function. And this is a notion from physics, but in our case, we assume that this function, this 1D function, is this isodepth function inside each domain. So each domain has a separate isodepth. And this sounds like a lot of math, but the reason this is all kind of necessary to spell out is that under these two assumptions, you can prove that your gene expression will actually be a piecewise linear function of these isodepths inside each domain. And so, okay, if you kind of ignoring all else, the takeaways from this is that we have this piecewise model that generalizes our original work Gaston, where we assume that there's only one isodepth, and it kind of generalizes a lot of existing domain identification methods, which assume that you have constant expression inside each domain. And that's equivalent to this isodepth function being a constant function inside each domain. And so with this kind of general setup, you can pose this problem, which we call this piecewise spatial topography problem, where you'd like to learn these spatial domains, these domain-specific isodepth functions, which define these kind of topographic maps inside each of your domains, and these linear 1D functions that best fit your data. And the way that we ended up doing this is through deep learning. So it turns out actually that instead of solving a very hard mathematical problem, you can just approximate everything with neural networks. And then this is actually equivalent to training a network that has a specific form in the deep learning literature that's called a mixture of experts model. And I won't get into details here, but at a high level, what you can imagine is that you have this routing network, which learns these different domains inside your tissue. And then you have what are called experts, which learn this isodepth function inside each of your domain. And so each of these experts is kind of a little copy of our Gaston algorithm, but we like kind of piece them all together. And so we put this together in a algorithm that we called GASTON-Mix, which uses the spatial mixture of experts to learn these kind of domain-specific topographic maps inside each domain of your tissue. And so first, we wanted to show that GASTON-Mix actually improves domain identification. And we found that when there are significant expression gradients, that we can actually get improved domain identification. So we kind of simulated a tissue where it follows this sort of checkerboard pattern, like the one from Jackson et al. That I'd shown before. And we model two different isodepths inside each domain, one that's kind of left to right, and then one that's like top to bottom. And we simulate noisy gene expression values according to these isodepths. And we find that if we compare to these kind of state-of-the-art domain identification methods, as well as our previous method, GASTON-Mix is more accurately able to identify these different like checkerboard domains. And that's because many of these existing methods for finding domains don't model gradients inside each domain. So they get kind of confused within a domain, while Gaston isn't able to model this like kind of intricate pattern. It gets kind of confused here. And then we also show that our algorithm more accurately learns spatial gradients, in particular that it more accurately learns these isodepths. And so, for example, compared to GraphSD, this is one domain identification method, which learns like these embeddings, but these embeddings don't really correspond to they don't correspond to a variable that lets you measure spatial gradients. And then Gaston gets kind of confused in these two parts of the domain, while GASTON-Mix can more accurately find this like kind of X coordinate. And then to show you this, it also works in real data. Here's one example of a Murfish data set. This is from the mouse anterior forebrain, which is this region of the brain that apparently has like many gene expression gradients. And we find that compared to the kind of existing methods, if you evaluate them using this assumed ground truth Allen Brain Atlas labels, Gaston-Mix is more accurately able to identify these different regions of the mouse anterior forebrain. And in particular, we were able to more accurately identify this like orange region, this like CP region, which often has lots of these gene expression gradients, and all of the other methods get kind of confused by. Like, for example, cell charter sort of separates this domain into like two separate ones. And in fact, if you dig a bit closer inside, is this showing up? Oh, yeah. And then if we look a bit closer inside this domain, we see that our algorithm learns this isodepth, this coordinate, which varies kind of from the left to the right. And so using this coordinate, you can look at different gene expression patterns. So for example, this gene CNR1, it has a gradient from the left to the right, and it has one of the largest slopes that our algorithm finds. And it's actually known to be a marker gene for the gradient inside this domain. And what's cool is that once you have this coordinate, you can also do kind of further downstream analyses. So for example, we found that this gradient of gene expression of this gene CNR1 is actually does not correspond to cell type. And if you look at different cell types, you still have this gradient even within different cell types. So it's sort of like a cell type independent gradient that could be due to maybe how you cut the tissue or neuron connectivity or other sorts of things. And then finally, we also applied our method to a tumor. Kind of the high level takeaway is that you can find these different domains, these different clones in a tumor. And using this, you can identify gradients that are both shared across different kind of clones of a tumor, and you can find domain-specific gradients. For example, domain 2, this kind of circular one, has a TNF alpha gradient, while domain 3 has like a gradient of oxygen, this hypoxia gradient. And so to summarize, we defined this new model of continuous variation of gene expression within spatial domains, which is broadly applicable to different spatial omics modalities. And we have this algorithm GASTON-Mix, which uses mixture of experts to learn both spatial domains and gradients inside each domain. The paper is kind of in bioinformatics now, so this might be outdated. We have code online. Then finally, I'd like to acknowledge my many helpful collaborators like Daisy, Fenna, and Ben, as well as the rest of my PhD lab, Boyang, for directing us to the MOE literature and our funding. And then as a shameless plug, I am starting a new lab in a month. And so if you find any of this interesting, I'm happy to chat.
Thank you very much. Questions? Yeah.
Thank you. That's a very nice talk. I was wondering how you determine the number of isodepth functions beforehand, like especially for real data. Is overfitting a problem here?
Yeah, that's a good question. That's one we actually still don't quite have the answer to. I think for these different applications, we used the number of like clusters found by another method, which seemed like a reasonable heuristic. But I think it's still open both how to find the number of domains, and also within each domain, do you want there to be a single isodepth, or maybe there's like kind of a radial structure and like different domains inside like one of these domains. I think these model selection questions in general are quite hard. So I don't know, choosing K and like K means is always like a tricky question. And there's like certain elbow heuristics that you can use. And we also have a heuristic that is not necessarily perfect. But yeah, good question.
Great. Thank you very much, Uthsav. Another round of applause.